Our third section of nervous tissue discusses membrane potentials and currents. This is the electrophysiology of neurons and how signals are generated. A nerve pathway is a series of separate cells. Neural communication is the production of electrical potentials and currents along that pathway of cells. An electrical potential is when we have different concentrations of charged particles in different parts of the cell. An electrical current is the flow of those charged particles from one point to another within the cell. Living cells are polarized. The resting membrane potential of neurons is negative 70 millivolts with a negative charge on the inside of the membrane. The resting membrane potential comes from the unequal electrolyte distribution between the extracellular fluid and the intracellular fluid. There's a diffusion of ions down their concentration gradients, and the plasma membrane is selectively permeable. There's also an electrical attraction of cations and anions. So many large anions are trapped inside the cell and therefore give the cell its negative charge. The membrane is very permeable to potassium, which will leak out until the electrical gradient attracts it back in. There's also cytoplasmic anions, negatively charged ions, that cannot escape due to their size or charge. These include phosphate, sulfate, organic acids, and proteins. The membrane is very permeable to sodium, and our sodium-potassium pump pumps out 3 sodium for every 2 potassium it brings in. This pump works continuously and requires a great deal of ATP. This is why glucose and oxygen must constantly be supplied to the nervous tissue. There's more sodium outside the cell and more potassium inside the cell. This difference creates our resting membrane potential. A local disturbance in membrane potential is called a local or graded potential. These occur when the neuron is stimulated by chemicals, light, heat, or mechanical disturbance. This depolarization decreases the potential across the cell membrane due to the opening of gated sodium channels. When these channels open, sodium rushes in, down the concentration gradient, and the electrical gradient. Sodium diffuses for a short distance inside the membrane, producing a change in voltage called a local potential. A local potential does not reach threshold, but simply deflects the resting membrane potential towards 0, depolarization, or away from 0, hyperpolarization. Local potentials are decremental. They get weaker the farther you get from the depolarization. You can see the red depolarized region and how that spreads out like a wave in a pond. A graded potential occurs in response to the opening of a mechanically gated or ligand-gated ion channel. When these ion channels open, they let in sodium or calcium, or they can let out potassium or let chloride in. So a number of different ions can flow different directions through these channels. A mechanically gated channel opens when pressure is applied and will let sodium and calcium in. An acetylcholine channel opens when acetylcholine is bound, and that is generally a cation channel. A ligand-gated channel that binds glycine will open up and it will allow chloride to pass. So all of these different channels can cause different molecules to move through the membrane. During a graded potential, the stimulus strength makes a difference. If we have a small stimulus, it will cause a small change in membrane potential. If we have a big stimulus, it will cause a large change in membrane potential and get us closer to threshold. Graded potentials or local potentials can be added together to become larger in amplitude. If we signal a cell a second time before it's recovered from the first signal, then it will be an additive response. So some differences between local potentials and action potentials. Local potentials are graded. This means they vary in magnitude with the stimulus strength. They are decremental. They get weaker the further they spread. And they are reversible as potassium diffuses out of the cell. These local potentials can either be excitatory or inhibitory. Next, we'll take a look at action potentials and how they differ from graded potentials.